Part 6 of carbohydrates. Let us see mutarotation with mechanism. What is mutarotation? The spontaneous change in specific rotation of solution of an optically active substance when standing to equilibrium value is known as mutarotation. So, for example, D plus glucose exists in two isomeric forms which undergo mutarotation, that is alpha D plus glucose and beta D plus glucose. In case of alpha D plus glucose, the specific rotation when ordinary D glucose is crystallized from water or acetic acid or alcohol water at temperature 30 degrees Celsius, then it forms alpha D plus glucose and it has melting point 146 degrees Celsius and specific rotation 112 degree, while beta D plus glucose has 150 degree Celsius melting point and specific rotation is of plus 19 degree. If either of the two forms is dissolved in water and form allowed to stand, then the specific rotation of solution changes gradually equilibrium, and that equilibrium value is 52.7 degree. The specific rotation of alpha D plus glucose gradually drops from initial value of 112 degree to 52.7, and specific rotation of beta D plus glucose gradually rises from initial value of 19 degree to 52.7 degree. The change in the rotation of each of these two forms to the equilibrium value, that is called as mutarotation. So what happened? Graphically we can represent that as time with the time, the mixture of alpha D plus glucose and beta D plus glucose at equilibrium have 52.7 degree, as alpha D plus glucose initially has 19 degree specific rotation, but its 19 degree increases to 52.7, and alpha D plus glucose plus 1 degree, it drops down to 52.7 degree. So beta D plus specific rotation of beta D plus glucose increases from 19 to 57.2, and 52.7, and specific rotation of alpha D plus glucose drops from 112 degree to 52.7 degree. To explain the phenomena of mutarotation, Tolan assigned an oxide ring structure to D plus glucose, and this is the ring structure. Glucose forms a stable cyclic hemiacetal by reaction between CHO group and OH group on carbon number 5. This carbon number 5, this OH and CHO, that is aldehyde group. Here reaction takes place as a result of cyclization. The C1 of glucose becomes asymmetric. Now this C1 becomes, this 5 is already asymmetric, but aldehydic CHO, this carbon is not asymmetric, but after cyclization it becomes asymmetric or chiral center, giving two isomers of glucose, that is 1 and 2. which only differ at this asymmetric center C1. Such a pair of diastereomers are called anomers, and this carbon is referred as anomeric carbon. So this this carbon and this carbon, these two carbons are called anomeric carbons because these two structures differ only at this carbon. The isomer having OH group right side of C1. So isomer, these two isomers are there, and here the OH group at right side of the this anomeric carbon, it is known as alpha D plus glucose, and the isomer where this hydroxy group is at left side of the anomeric carbon, then it is known as beta D plus glucose. The mutation occurs due to slow interconversion of D plus glucose to beta, alpha D plus glucose to beta D plus glucose via the open chain form until the equilibrium is established, giving constant specific rotation of 52.7 degree. The equilibrium is attained faster in presence of acid and base. And the alpha D plus glucose and beta D plus glucose are readily hydrolyzed by water in aqueous solution. Anomer is converted via open chain form into an equilibrium mixture containing both cyclic isomers. At equilibrium, it contains 64% beta form and 36% alpha form, and the concentration of open chain structure is very low, that is 0.01%. So beta form is 64%, alpha form is 36%, and open chain structure it is 0.01%. So mechanism of mutarotation. Lowry in 1925 suggested the mechanism which involves simultaneous attack by acid and base. He showed that the mutarotation is only possible in presence of amphoteric solvents such as water. Mutarotation is arrested in pyridine base or cresol acid, but it takes place in pyridine and cresol, mixture of pyridine and cresol. Mechanism involves simultaneous addition and elimination of proton following concentrated mechanism, following concerted mechanism having third order kinetics. So what is meant by concerted? This concerted is nothing but it is cyclic, or we can say continuous. There are no steps that first step, then second step. All steps occur simultaneously. This type of mechanism is known as concerted type of mechanism. See, it involves simultaneous attack by acid Ha or H2 and a base B minus or H2 yield an open chain structure or open chain aldehyde form D plus glucose, which then recyclize to give other form. Here, how this alpha D plus glucose convert to open chain, and this open chain can convert to beta D plus glucose, and vice versa. Beta D plus glucose convert to open chain structure, and this open chain structure can convert to alpha D plus glucose. It's shown in this mechanistic path. So alpha D plus glucose, when the proton, that is OH proton, is abstracted by base, and here conversion of this group into aldehydic group takes place. What happen as this OH bond breaks and there is formation of carbonyl, this bond also breaks and this O minus O minus abstract proton from acid and we get here CHOH. So here see this one is CHOH and this aldehyde. This is open chain structure of D plus glucose. And this open chain structure of D plus glucose can be easily converted into beta D plus glucose. Similarly, beta D plus glucose when react with base, base abstract proton of hydroxy group on anomeric carbon and this hydroxy groups converts into carbonyl C double bond O that is aldehyde group. This cyclic structure breaks into acyclic and this O minus abstract proton from acid and we get CHOH. This is beta and this beta D plus glucose converts to open chain structure as this one. So via the open chain structure, alpha can convert into beta and beta can convert into alpha. But the mechanistic path goes via open chain structure. Thank you.